Meiosis, on the other hand, is used for just one purpose in the human body, the production of gametes, sex cells or sperm and eggs. Its goal is to make daughter cells with exactly half as many chromosomes as the starting cell. To put that another way, meiosis in humans is a division process that takes us from a diploid cell, one with two sets of chromosomes, to haploid cells, ones with a single set of chromosomes. In humans, the haploid cells made in meiosis are sperm and eggs. When a sperm and an egg join in fertilization, the two haploid sets of chromosomes form a complete diploid set, a new genome.
Phases of meiosis. In many ways, meiosis is a lot like mitosis. The cell goes through similar stages and uses similar strategies to organize and separate chromosomes. In meiosis, however, the cell has a more complex task. It still needs to separate sister chromatids, the two halves of a duplicated chromosome, as in mitosis. But it must also separate homologous chromosomes, the similar but non-identical chromosome pairs an organism receives from its two parents. These goals are accomplished in meiosis using a two-step division process. Homologue pairs separate during a first round of cell division, called Meiosis I. Sister chromatids separate during a second round called Meiosis II. Since cell division occurs twice during meiosis, one starting cell can produce four gametes, eggs or sperm. In each round of division, cells go through four stages: prophase, metaphase, anaphase, and telophase. Meiosis I. Before entering meiosis I, a cell must first go through interphase. As in mitosis, the cell grows during prophase I. Differences from mitosis begin to appear. As in mitosis, the chromosomes begin to condense, but in meiosis I, they also pair up. Each chromosome carefully aligns with its homologue partner so that the two match up to corresponding positions along their full length. This process, in which homologous chromosomes trade parts, is called crossing over. It's helped along by a protein structure called the synaptonemal complex that holds the homologues together. The chromosomes would actually be positioned one on top of the other, as in the image below, throughout crossing over. They are only shown side by side in the image above so that it is easier to see the exchange of genetic material.
In Anaphase I, the homologues are pulled apart and move apart to opposite ends of the cell. The sister chromatids of each chromosome, however, remain attached to one another and don't come apart. Finally, in telophase I, the chromosomes arrive at opposite poles of the cell. In some organisms, the nuclear membrane reforms and the chromosomes decondense, although in others, this step is skipped. Since cells will soon go through another round of division, cytokinesis usually occurs at the same time as telophase I, forming two haploid daughter cells. Meiosis II. Cells move from meiosis I to meiosis II without copying their DNA. Meiosis II is a shorter and simpler process than meiosis I, and you may find it helpful to think of the meiosis II as the mitosis for haploid cells. The cells that enter meiosis II are the ones made in meiosis I. These cells are haploid, have just one chromosome from each homologue pair, but their chromosomes still consist of sister chromatids. In meiosis II, the sister chromatids separate, making haploid cells with not duplicated chromosomes. During prophase II, chromosomes condense and the nuclear envelope breaks down if needed. The centrosomes move apart, the spindle forms between them, and the spindle microtubules begin to capture chromosomes.
Two sister chromatids of each chromosome are captured by microtubules from opposite spindle poles. In metaphase II, the chromosomes line up individually along the metaphase plate. In anaphase II, the sister chromatids separate and are pulled towards opposite poles of the cell. In telophase II, nuclear membranes form around each set of chromosomes and the chromosomes decondense. Cytokinesis splits the chromosome sets into new cells, forming the final product of meiosis. Four haploid cells, in which each chromosome has just one chromatid. In humans, the product of meiosis are sperm or egg cells. Does meiosis always produce four gametes? How meiosis mixes and matches genes? The gametes produced in meiosis are all haploid, but they're not genetically identical. For example, take a look at the Meiosis II diagram above, which shows the product of meiosis for a cell with 2n equals 42, n equals 42. N equals 4 chromosomes. Each gamete has a unique sample of the genetic material present in the starting cell. As it turns out, there are many more potential gamete types than just the four shown in the diagram, even for a cell with only 4 chromosomes. The two main reasons we can get many genetically different gametes are crossing over. The points where homologues cross over and exchange genetic material are chosen more or less at random, and they will be different in each cell that goes through meiosis.
If meiosis happens many times, as in humans, crossovers will happen at many different points. Random orientation of homologue pairs. The random orientation of homologue pairs in metaphase 1 allows for the production of gametes with many different assortments of homologous chromosomes. In a human cell, the random orientation of homologue pairs alone allows for over 888 million different types of possible gametes. When we layer crossing over on top of this, the number of genetically different gametes that you or any other person can make is effectively infinite.